Hi everybody, my name is Aderisola Adikwe, and I am going to be talking about microscopes. Not the microscope itself, but like the processes that a tissue has to undergo before it can be properly examined under a microscope. So we're going to be talking about histology. Histology is the study of cells of tissues under a microscope. So, but before a tissue can be fully or properly understood and studied under a microscope, it has to go through seven processes. And I summarize the processes into a mnemonic called Falake Daniels can eat 7 meals simultaneously. So the stands for fixating, D stands for dehydrating, the the C stands for clearing, the E embedding, the S for sectioning, for mounting it on the slide, and then the last S for staining. But like we have to understand what each of these things are, because even if somebody tells you, oh, ensure to fixate, ensure to dehydrate, ensure to clear or to embed, you can't fully grasp that until you know what that is. So when you enter the lab, what should you do? So the first step we talked about was fixating, right? So fixating has to do with adding of fixatives. And what are fixatives? Fixatives are substances that either preserve like the structure of the tissue, preserve the structure, and they stop all ongoing chemical processes that are occurring in the tissue. But what fixative is commonly used? The fixative commonly used is formalin. So formalin is used, and when you ask yourself what is formalin, formalin is a 10% formaldehyde. So when you want to fixate, you use formalin, which is a 10% formaldehyde. Like the second step is dehydrating. Dehydrating just simply means what it is, removal of water. When you go for a run and you're like, oh, I'm dehydrated or something like that. Or it's very easy to remember because the way you remove water is the same way water leaves the body for people that drink a lot of alcohol. So to fixate a tissue, you use alcohol. So you incorporate alcohol into the tissue progressively. So you start from like a low percentage alcohol, like 30% alcohol for 15 minutes. Then you do 50% alcohol, then you do 70% alcohol, and you keep adding until you get to like real or pure alcohol, which is like 98.9% to 100% alcohol. So that's the second step. Basically, you dehydrate by adding alcohol. Now alcohol is sort of like a use and dump, like if you're talking about that type of like gutter language, but like a use and dump type of thing, because the next step is called clearing, and clearing just means the removal of the alcohol from the tissue. So you use the alcohol to dry it out. Now you have to remove the alcohol. And how do you do that? You should keep the tissue inside the substance that is soluble in paraffin. So this substance that is used for clearing most times is called xylene, which is Xylene, or chloroform. I think chloroform is something that I've heard for a really long time. So to me, it's more popular. But the one that is commonly used in the process of clearing is xylene. Yeah. So the next step is called embedding. Embedding means the addition of a substance so that the tissue becomes rigid, the tissue becomes fixed. So how do you do that? You use a substance, like we said, for fixating, you use formalin, which is 10% formaldehyde. For dehydrating, you use alcohol. For clearing, you use a substance soluble in paraffin, which is xylene or chloroform. The next step tells you why you have to use a substance soluble in paraffin, because to embed, to make the tissue rigid, you need to use paraffin wax. So when paraffin wax is heated to 60 degrees Celsius, it actually melts. So you use paraffin wax in its melted form to to make the tissue rigid. And because the tissue is now rigid, it is now able to be sectioned. So a very important step is sectioning. Sectioning has to do with cutting the tissue into smaller pieces, and it is done using a machine called microtome. So a microtome is basically a machine that has glass knives. Like I just feel like it's so cool. So like it has glass knives, and it basically cuts the tissue down into like 3 to 10 micrometers to allow it to be like easily viewed under the microscope. So after embedding, we have mounting it on the slide. So that's like so literal. You take your slide and you put the sectioned part tissue on it. And the next thing you do is staining. Staining is a technique that is like commonly done with like two substances, either hemotoxylin or eosin. So hemotoxylin stains protein as red, and eosin stains protein as pink. So it's basically a staining technique. And then the last technique, before you can say, okay, I'm ready to put this on my microscope and examine, is called, it's not actually a named technique, it's just to cover up the slide with the cover slip. So after that, you're ready to you're ready to examine your tissue. So remember, Falake Daniels can eat 7 meals simultaneously. There are 7 steps in preparing your tissue for examination. The first step is fixating. The second step is dehydrating. The third step, clearing. Fourth, embedding. The fifth, sectioning. The sixth, mounting it on the slide. And the seventh, staining. Fixating is done with formalin, FF. Dehydrating is done with alcohol. Alcohol makes you dehydrated. Clearing is done with xylene and chloroform. Embedding is done with paraffin wax at 60 degrees Celsius. Sectioning is done using a microtome, which is a machine that has glass knives. Mounting on a slide is done using your hands, just mount them on the slide. And then, well, maybe use your gloves, I don't know. You mount them on your slide. And then the last step is staining, which is done using hemotoxylin or eosin. Thank you for watching. Hope you understand. Bye.